When viruses replicate, they must produce viral proteins and reproduce their genomes. That's how they spread. The information for these processes is coded for in the viral genes. Normally, RNA viruses contain about 10 genes, while DNA viruses, such as herpes virus, can contain about 80 genes. Viral genes can be divided into those that code for structural proteins and non-structural proteins. The structural proteins actually make up the body of the virus, such as the subunits that build the capsid. Other structural proteins can directly associate with the viral nucleic acid. Spike proteins that sit within the viral envelope, for example, are also structural proteins. The non-structural proteins, on the other hand, are the enzymes needed to produce viral genomes, or viral messenger RNA. Now, this is especially true for RNA viruses that require unique enzymes, polymerases, which are able to produce RNA genomes off of RNA templates. All these synthetic processes go on inside the host cell's cytoplasm. So the very first step in viral infection, the virus's initial task is to select and get into the right host cell. For this, the viral spike protein must bind to a susceptible host cell, a cell that presents on its surface a cell protein that serves as a specific receptor for the virus. Frankly, this seems a bit absurd. Why would a cell present virus receptors on its surface? Actually, they don't. The cell has many proteins on its surface, and they all have functions important to the cell for its survival, or it enables the cell to do its job in the body. Viruses have evolved to exploit and take advantage of these proteins and use them as holds or handles to latch onto and bind specifically. Think about it. When we examine the surface of a natural rock formation, El Capitan in Yosemite, for example, we see nooks and crannies, crevices and pockets in the rock. Clearly, these don't exist for the pleasure of rock climbers. However, the climbers exploit these structures as holds, places they can latch onto, grasp, and gain footage to ascend the rock and reach its summit. Viruses do the same. They view the cell surface proteins as holds and use them as virus receptors that allow them to bind and gain entrance into the target cell. Thus, the presence of receptors recognized by the virus is what makes a cell a viable target for viral infection. Having a receptor makes a cell susceptible and defines the specificity of a virus for its host. Once bound, the virus must enter the cell, produce its proteins, replicate its genome, and release a new generation of infectious particles. Sounds complicated? I agree, so bear with me. Let's take it now step by step. First, the bound virus must cross the cell membrane. For enveloped viruses, this entails fusing the cell membrane with that of the virus. Then, the capsid containing the viral genes breaks down and releases its genome into the cytoplasm. When the viral genome is plus-stranded RNA, it can serve as a messenger RNA immediately and be translated to produce all the viral proteins, structural and non-structural. Viral polymerases can then produce multiple copies of the viral genome. These components next must assemble into new viral particles comprised of capsids containing viral genomes and other viral structures. As the particle emerges from the cell, it may take on an envelope derived from the cell membrane. The newly formed virus particles can then move on to neighboring cells, and if these are susceptible, they too can get infected, and thus a new generation of infectious viruses is born, ready to spread and cause disease in others.